Okay, so we're going to get started. It's my absolute pleasure and joy to introduce Dr. Laura Cheney from Albert Einstein College of Medicine. Laura is an infectious diseases physician scientist par excellence and truly a rising star. She's an assistant professor in medicine and pathology. And actually, her passion for HIV research dates back to college, where she was at Emory University and did her first foray into HIV research at the CDC. She then did her MD PhD at Stony Brook, took 6 months and went to Botswana for field work in HIV, and then did a PhD on the role of NEF on inflammation and I think on metabolism, but I'm not positive about that. The important point is also that she's a recipient of a K award, a KO8, which is very challenging to get. And she came to my lab, and I have the privilege of being her mentor when we were just sort of learning a little bit about autophagy. And Laura really has spearheaded and taken over the study of autophagy and its role in HIV-NCI and moved it into really new arenas. And she's going to talk to us today about that. So I look forward to you all enjoying her talk and hearing about her new and very important translational findings. So Laura, go ahead.
All right. Thanks, Joan, for the introduction, and thank you everyone for being here today and for giving me the opportunity to talk about this project, this research program that I've been building that I'm extremely passionate about, and I hope to impart some knowledge to you all today. Hmm, there we go. I've got nothing to disclose. So I don't need to tell anybody in this audience really how impactful antiretroviral therapy has been for the lifespan and health span for people with HIV. But despite this, comorbidities associated with aging occur prematurely. This, of course, includes HIV-associated neurocognitive impairment, or HIV-NCI. 15 to 50% of people with HIV develop HIV-NCI despite controlled viremia. HIV-NCI is a waxing, waning spectrum of behavioral, motor, and cognitive abnormalities, significantly impacts the quality of life for people with HIV, and remains an independent risk factor for mortality even in this current ART era. It's challenging to diagnose, and there's no specific treatment for people who take ART. So the pathogenesis of HIV NCI is complex and multifactorial. And that it persists in the current ART era suggests that ART may actually contribute to HIV NCI. There are clinical studies that have found some associations between NCI and specific antiretroviral drugs, drug classes, duration of our exposure. And on the lab side of things, antiretrovirals of all types have been shown to be toxic to different types of CNS cells. So it's probable ART plays a role in HIV NCI development. But the mechanisms by which ART may cause cell toxicity and contribute to NCI are not completely characterized. A more complete understanding of the contributions ART makes to NCI may lead to development of therapy for NCI in people with HIV. So I'm interested in further characterizing the contributions ART may make to HIV-NCI by their impacts on astrocytes. Astrocytes are really abundant cells in the brain, dare I say, underappreciated cells in the brain. And they're key homeostatic cells with many really important functions. I have here on the left some global functions they serve, like maintaining blood-brain barrier integrity, participating in immunity. And on the right is some functions they serve in the CNS microenvironment. I highlight that, or I just want to highlight that glutamine and glutamate shuttling and the antioxidant response are known to be abnormal in people with HIV. So with all that they accomplish in the CNS, it's probably easy to see that if these functions are disrupted, so is proper CNS functioning. So one very important intracellular process that is important for astrocytes to maintain their homeostasis, and then by extension CNS homeostasis, is autophagy. Autophagy is a highly regulated intracellular proteolytic process. It's depicted here at the bottom. Damaged organelles, protein aggregates, other cytoplasmic garbage are all enclosed within a specialized double membrane vesicle termed an autophagosome. This then fuses with a lysosome, and everything inside is then degraded by lysosomal enzymes. And the building blocks for energy and other cell processes can be recycled back out for use by the cell. Autophagy is essential for cell metabolism and quality control. Dysregulation of any step in this process can contribute to neurodegenerative conditions, which I'm highlighting now. Defects in autophagy have been identified in steps in autophagy from start to finish in Parkinson's, Alzheimer's, ALS, frontotemporal dementia, Huntington's, you name it. So given the importance of autophagy to astrocytes, we asked if antiretroviral drugs may be contributing to HIV-NCI by interfering with autophagy in astrocytes. There is literature to support this question. These tables are from two separate reviews we wrote in which we focused on what is published regarding the effects of antiretroviral drugs on autophagy in any cell type and disease process. That's that table on the left, or repositioning them as cancer chemotherapy through their effects on autophagy. That's the table on the right. I have zero intention of making you read that teeny tiny script. I just wanted to show these tables to highlight that there are only 64 studies that examine ART in this context of autophagy, many of them not even related to HIV and its comorbidities, and that there are even fewer studies that were focused on HIV-NCI. These are highlighted in the red boxes. So these tables are meant to just show you that ART is doing something to autophagy, but that also there isn't not much known about what that something is, especially regarding astrocytes and contemporary antiretroviral drugs. So I'm arriving at my hypothesis, which is that ART inhibits astrocyte autophagy, resulting in disrupted homeostasis and subsequent neuronal dysfunction, contributing to HIV-NCI. And our ultimate objective is to have a better understanding of autophagy changes in the context of HIV-NCI, which could then drive drug development for antiretrovirals with less off-target effects, or maybe develop targeted autophagy modulators to mitigate HIV-NCI. We first determine whether ART inhibits autophagy in astrocytes, and spoiler alert, it does. For this, we treated primary human astrocytes for 24 hours or daily for 7 days with a combination of tenofovir, emtricitabine, and dolutegravir. This is a relatively contemporary treatment regimen for people with HIV, and I used concentrations consistent with average cerebral spinal fluid levels of each of these drugs. The 24-hour time point was chosen for typical autophagy dynamics after a stimuli, and the 7 days of daily treatment is meant to mimic a patient's experience, taking their antiretroviral drugs daily long-term. In the last 2 or 4 hours of ART treatment, I added chloroquine to inhibit lysosome degradation, collected lysates for Western blotting, then to assess 2 markers of autophagy, LC32, which is the autophagosome marker, and P62, which assists in cargo selection. So LC32 associates with autophagosomes from start to finish, making it the autophagosome marker. It studs both the outer and inner membrane of the autophagosome. Autophagy is a constitutive process such that autophagosomes are constantly being formed and then degraded. So this constitutive process, LC3, is constantly being made, and then whatever is present on the inner autophagosome membrane is constantly being degraded when the autophagosomes or APG fuse with lysosomes. The Western blotting for LC32 is commonly used to measure autophagy dynamics. But because autophagy is dynamic, any change in LC32 after treatment could represent a decrease in production or an increase in degradation. So to tease out the specific effects of treatment on autophagy, we use a chemical inhibitor of degradation, that's chloroquine. Chloroquine inhibits the acidification of the lysosome, thereby preventing the degradation. Now, LC32 with chloroquine accumulates and therefore represents only production. So without and with chloroquine, we can determine both LC32 production and degradation after treatment relative to control. P62, not shown in this picture at the top there, also associates with the autophagosomes from start to finish and is also degraded inside the lysosome when the APG fuse. And so Western blotting for P62 is commonly used to confirm the LC32 Western blotting data. One more slide, I promise, before I get to data. I don't want to lose anybody with terminology. So just to be clear, autophagosome formation, which correlates with LC32 production, is really called biogenesis. And flux, which is really the term used to describe this entire process from start to finish, often is used to denote cargo degradation. So these are the terms that are going to be on the graphs on the next slide. So finally, some data. After 24 hours, shown on the top, and also after 7 days of daily ART, shown at the bottom, we find that autophagy is inhibited in primary human astrocytes. Here are representative Western blots for LC32 and P62. You can see there's a baseline amount of LC3 produced, and then the accumulation of it in the presence of chloroquine. You can see this too for P62. So the graphs are derived from the optical densities from the blots, and the graphs show full changes in biogenesis and flux over control, represented by the dashed lines at 1 for both 24 hours, again on the top, and 7 days daily ART treatment on the bottom. So we show that biogenesis, autophagosome formation, as well as degradation, labeled flux on these graphs, are decreased at both time points, indicating ART inhibits autophagy. P62 production is entirely different from LC32, so there's no biogenesis on those graphs. But since P62 is degraded along with LC32, we can tease out P62 flux after ART treatment as well. And it is also significantly decreased at both time points. This supports the LC32 findings that ART inhibits autophagy in the astrocytes. So these data show ART inhibits autophagy and may do so at different steps, which is really interesting. Biogenesis and degradation. This inhibition is problematic in and of itself, and over time may disrupt CNS homeostasis, contributing to HIV-NCI. Other questions stemming from this data are, what are the mechanisms by which ART is causing this inhibition? And also, a lot of cell processes are regulated in part by autophagy. So are these other cell processes affected? And if so, how are they affected by ART-induced autophagy inhibition? Also, is selective autophagy inhibited? I'm going to focus on selective autophagy, specifically mitophagy, for the rest of the talk. Selective autophagy is targeted degradation of specific or selected intracellular cargo in the autophagic system. Selective autophagy is mediated by autophagy receptors and adapters that recognize specific cargo and then are able to incorporate that cargo into forming autophagosomes. One autophagy adapter is P62, which predominantly recognizes polyubiquitinated proteins, including polyubiquitinated mitochondria. Cells need to maintain a healthy population of mitochondria for energy, also survival. Damaged and dysfunctional mitochondria play a significant role in cell fate. It can tip the scales towards cell death. So it behooves cells to have some mechanism to dispose of irreparably damaged mitochondria. Mitochondrial fission and fusion are important mechanisms for this, but mitophagy also contributes to maintaining that healthy population of mitochondria. Mitophagy is the selective recognition and degradation of mitochondria in the autophagic pathway. This helps maintain homeostasis in cell survival. It's vital. Previous publications have shown that antiretrovirals can impact mitophagy, and I just showed you data that P62-mediated selective autophagy may be inhibited by ART. If mitophagy is inhibited by antiretroviral drugs, this could potentially lead to loss of homeostasis and cell death, but the effects of ART on mitochondria integrity and on mitophagy in astrocytes are not known. So to determine if ART is causing mitochondrial toxicity, which would be a stimulus for mitophagy, we examined a marker of mitochondrial toxicity, which is membrane polarization. Mitochondrial membrane polarization, it's the charge difference between the inner and outer mitochondrial membranes. ATP generation is dependent on that charge difference. So damage to either the inner or outer membrane can lead to depolarization, and then ATP is not generated, which would be a stimulus for mitophagy. So cells were treated for the 24 hours or daily for 7 days with ART, and then polarization was measured by fluorimetry. Treatment with CCCP for 1 hour was also included as a positive control. CCCP disrupts that proton gradient between the inner and outer membranes, causing depolarization. So these graphs show the percent of polarization relative to control, shown by the dashed line at 100. There's a significant decrease in membrane polarization after 1 hour of CCCP. We expect this. And we find no difference in polarization after either 24 hours or 7 days of daily ART. LDH assays were performed in conjunction with these to assess general cytotoxicity, and neither 24-hour or 7 days of daily ART caused significant cytotoxicity relative to the maximum amount of LDH that could be released from a set of lysed cells. Another marker of mitochondrial toxicity is mitochondrial ROS production. I'd love to show you mitochondrial data related to mitochondrial ROS production, but after exhaustive troubleshooting, I could not reliably detect mitochondrial ROS. Instead, I'm showing general ROS production after 24 hours and 7 days of daily ART treatment. The graph is the percent of ROS at either time point relative to control set to 100. ART does not cause any significant change in ROS at either time point. We also performed Western blotting for SOD2, which is a superoxide dismutase specific to the mitochondrial matrix and is responsible for converting superoxide into hydrogen peroxide within the mitochondria. This graph is the full change of ART relative to control set to 1, And there's no difference in SAW2 at either time point. So if ART were causing mitochondrial ROS, the astrocytes could probably neutralize it, which would be really good for homeostasis. Well, I don't think ART is toxic to the mitochondria, at least not in the ways I so far examined it, which is good. So from that perspective, mitophagy shouldn't be impacted. However, I showed you that ART inhibits autophagy. P62-mediated autophagy is also inhibited. So regardless of whether ART is toxic to mitochondria or not, ART still could be impacting mitophagy. I'm examining mitophagy with a fluorescent reporter, which I'm going to get to at the end. I've performed many complementary techniques to the reporter assay to examine flux. I'm going to go over that first. I promise I'm not burying any lead here. It's just the reporter experiments aren't complete yet. And I just, for fun, wanted to show an EM image I took for a previous experiment a long time ago. But I'm just showing you a pretty large autophagosome that's got mitochondria engulfed on the inside. Mitophagy is happening. So first I looked at mitochondrial mass. I treated cells with ART for 24 hours or daily for 7 days and measured mitochondrial mass with MitoTracker Deep Red by flow cytometry. If mitophagy was induced by ART, I'd expect there to be less mass. And vice versa, if mitophagy was inhibited by ART, I'd expect for there to be more mass. Here are representative histograms for either time point, and the graph shows the mean fold change of mean fluorescence intensity over control set to 1, and there's no significant change in mitochondrial mass at either time point. I also performed Western blotting for cytochrome C oxidase subunit 2. This protein is part of the electron transport chain and resides in the mitochondrial matrix. Matrix proteins are degraded in mitophagy. So if mitophagy was inhibited, I'd expect to see an increase in MTCO2. Here are representative Western blots for each ART time point. And this graph is the full change of MTCO2 relative to control. There's no change with ART at either time point. So by Western blotting, I also looked at proteins involved in mediating autophagic degradation of mitochondria, PINK1 and Parkin. These two proteins mediate the best understood pathway for ubiquitin-dependent mitophagy. PINK1 Parkin are activated in the setting of mitochondrial membrane depolarization. Here we have representative Western blots, and the graphs are the mean fold change over control, and we find no change in either protein at either time point. And while negative data, it actually corresponds nicely with our mitochondrial membrane polarization data, also negative. And also by Western blotting, I looked at amounts of BNP3L-NIX. This is another protein involved in mediating mitochondrial degradation in the autophagic system. BNP3L-NIX mediates mitophagy in a ubiquitin-independent manner. It can bind directly to LC3 to target mitochondria for degradation. So while there's no change in total BNP3L-NIX, there is a trend at 24 hours and a significant decrease after 7 days daily ART of BNP3L-NIX homodimer. This is really interesting. It was shown just a couple of years ago that homodimerization of BNP3L-NIX is important for mediating mitophagy. If it can't homodimerize, mitophagy is severely inhibited. So, so far, this data is the only hint that mitophagy may actually be inhibited by ART. And we didn't set out to look at mitochondrial fission and fusion, but we may incidentally learn that that might be impacted by ART as well. I had attended an Autophagy conference this past fall, and I learned that the decrease in the homodimerization might be interfering with mitochondrial fission and fusion, because this protein is involved in that process as well. So if so, this too could be contributing to the pathogenesis of HIV-NCI. So I've come now to the reporter assay that I mentioned before, which is the technique I'm using to definitively measure mitophagic flux. As I said, this is in progress, so I don't have any data. That's why I put this at the end. But I'd like to share the technique I'm using because I think it's pretty cool. Based on a relatively newly developed protein tagging system called HaloTag, I designed a custom mitophagy reporter to track mitochondria within the autophagic system. The reporter plasmid has a mitochondrial targeting sequence fused to Halo. Halo is a modified bacterial enzyme that forms covalent bonds with their ligands. The bonds are acid and protease resistant, which is perfect for tracking things that wind up in the very acidic, protease-rich environment inside the lysosome. And the ligands are conjugated to detection molecules like fluorophores. This is just a schematic that depicts how HaloTag protein labeling works. You have a plasmid with a gene of interest genetically fused to the Halo enzyme sequence. It's expressed inside cells. Then the ligand for Halo is added. The ligand is composed of the Halo enzyme substrate linker conjugated to a detection molecule. In my figure here, I just have GFP. Once the ligand is supplied, Halo attached to your protein of interest covalently binds to that ligand, and then your protein of interest can be detected by, for example, microscopy, looking for that GFP signal. So here's my experimental design. I transduce cells with the reporter plasmid and then treat daily for 7 days with the ART. We're only doing 7 days here because this is the time point at which we saw a change in the BNAB3L mix homodimer. Then provide the ligand for Halo that's coupled to a fluorophore. I will then label with LAMP1, which is a lysosome marker. The antibody is a fluorescence conjugated antibody for LAMP1. And then I'm going to perform microscopy to measure mitophagic flux. With this, the more mitochondria that co-localize with lysosomes, the more mitophagic flux is occurring, and vice versa, the less mitochondria co-localizing with lysosomes, the less mitophagic flux is occurring. So here's this HaloTag in action so far. The picture on the left is demonstrating that astrocytes express the HaloTag fusion protein in the right cell compartment. Here, there's no ligand present. Instead, I performed immunofluorescence for the Halo enzyme. That's what's shown in green. And immunofluorescence for TOM20 shown in the magenta color. TOM20 is an outer mitochondrial membrane protein. So this image shows that astrocytes are expressing Halo and that Halo is appropriately localized to mitochondria. The second picture here on the right is astrocytes transduced with the Halo reporter. I again performed immunofluorescence for Halo. That's shown in the green in the top and the bottom here. So both of these cells are expressing Halo shown by the immunofluorescence. Then I added the Halo ligand, which is conjugated to a red fluorophore. I added it to cells shown on the top, but not shown on the bottom. And you can see that the ligand is bound to Halo in red, and that red fluorescence is absent when there's no ligand present. So this confirms that the Halo enzyme is working, binding with its red fluorescent tagged ligand. So the next steps are to ensure responsiveness of the reporter to mitophagy modulators, and which will include the LAMP1 co-localization, and then actually perform experiments with antiretroviral drugs. To summarize, ART may differentially affect autophagic pathways in astrocytes such that non-selective autophagy is inhibited. That's the LC3-2 data. P62-mediated autophagy, one form of selective autophagy, is also inhibited. The jury's still out on mitophagy. Of the data that I've shown, there's just one hint that mitophagy may be inhibited, and that's with the decreased BNIT3L mix homodimerization. Regardless, a change in autophagy dynamics certainly impacts homeostasis of cells and long-term may contribute to HIV-NCI development. And other ongoing studies I have related to this work are mechanisms by which ART inhibits autophagic flux, and then, of course, other cell processes affected by the ART-induced autophagy inhibition. And I might turn some attention to this fission fusion story, kind of a interesting aspect, I think, that we could explore more intently. So these studies aim to understand better how HIV-NCI develops in the current ART era, specifically how autophagy, a really important cell process needed for homeostasis, is impacted by antiretroviral drugs. Effects on autophagy, particularly to CNS cells, should be an important consideration during new antiretroviral drug development. For those infected with HIV because of their risk for developing neurocognitive dysfunction, and also for those trying to prevent HIV infection with PrEP, I think this is particularly important as PrEP uptake continues to expand. Well, we ultimately aim to identify areas for NCI therapy development, such as to perhaps design autophagy modulators to counteract antiretroviral effects. This concept of directly manipulating autophagy for therapeutic benefit is already being applied in chemotherapy development and also in the neurodegeneration field. Or continue to improve antiretrovirals themselves to improve neurocognitive outcomes for people with HIV. And also, I mentioned possibly prevent neurologic side effects in those taking antiretrovirals to prevent HIV infection. So I just want to thank my amazing mentor, Joan, and our amazing lab members. I love coming to work every day. We work with such amazing people in our group. And a quick shout out to Grace McDermott, who's a medical student here at Einstein, who did a lot of the mitochondrial mass work. She's extremely talented, and I wish I could have kept her. Also, Dr. Ana Maria Cuervo, my co-mentor on my K. And I have an extremely supportive, just warm, amazing Einstein community. As I said, I love coming to work every day, and Einstein is home for me. And lastly, my funding, some of this was completed with a CFAR grant, and of course, a big thank you to NIMH for providing me with my K. So with that, thank you. I'll take some questions if anybody has any.
Thank you so much, Laura. Really exciting and new arena. And again, it just shows the importance of training people early. You know, she learned about HIV in college, continued to do it through her MD, PhD, her training, her residency, her fellowship, now her K, and will soon go on to independence and turn this into a major research project. And she's training other medical students and graduate students too, which is really so wonderful and part of this process. So do we have any questions?
I see that Raha's hand is up.
Okay, go ahead, Raha.
Oh, no, she's
I was
clapping.
just
My mistake.
applauding.
Sorry.
That was a really impressive presentation, but I think
thank
Scott
you.
has a question.
Thank you very much. Please, Scott.
So very nice presentation. I agree. Excellent work, and congratulations on your K and moving
Thank you.
forward. You're a physician, and I'm an infectious disease physician. I have a bit of a focus on pharmacology, but I see Jay McLaughlin in the audience today, so have to be careful about what I say. But when we talk about ART, as there's all sorts of ART.
Yeah.
And so, and in particular, the two formulations of tenofovir result in markedly different intercellular concentrations of tenofovir diphosphate. So I know this is early in your career, but I want to encourage you, of course, and I suspect you know this, to look at individual drugs in addition to those in combination, and there's substantial inter-individual and intra-individual variation in drug concentrations. And so to also look at different concentrations so that you get more of a dose effect, or get a dose effect, and can comment on whether or not that supports causality. So, your experimental conditions, of course, support causality, but even more so, varying the dose and looking at individual drugs would really further strengthen what you're doing. But I know that adds time and expense to experiments, particularly with the limited funds from a K award.
Yes,
So,
thank you. Thank you for that input. I will say that I did polarization assays and general ROS assays with a stable concentration. I didn't vary, and I don't see any change with the concentration that I had used. We are a little bit hesitant to go substantially higher in the concentrations just because we're really trying very hard to recapitulate what a patient might experience with their fixed doses of drugs. But I agree with you. I think that it's very important to tease out sort of what the range of effects could be, because as we both know, everybody knows there's intra and inter variability in the PK/PD situation with the drugs. As efaviren's probably being the best example of that.
So I see Dr. Bendayan here as well. So sorry for not calling her out as another pharmacology expert. So I'm sure she'll have some insights as well.
Thanks, Scott. Okay, Dr. Bendayan, since he already introduced you.
Thank you. Thank you very much, Scott. And Laura, thank you for this very nice talk. In fact, we've been having similar interest. We have not worked with astrocytes, but we have been looking at toxicity of ART at the level of blood-brain barrier models. Primarily, our work has been performed in human and mouse brain microvessel endothelial cells, as well as isolated capillaries, and particularly for dolutegravir. So our studies in general have been performed with just the integrase inhibitors, primarily as sole compounds. So maybe to better delineate the effects. And in fact, we've demonstrated that dolutegravir exerts inflammatory effects, these associated with ER stress mechanisms. And we have also looked more recently at autophagy and demonstrated similar effects as yours in the brain microvessel endothelial cells. So I just want to indicate that this is really interesting, certainly a growing field. And it's interesting that comparing to bictegravir, we see a much safer profile with bictegravir in terms of neurotoxicity compared to dolutegravir. So my question to you is that obviously you worked with the drugs in combination. How can you delineate, or can you, the effects? I mean, in terms of the combined ART, and now as I said, we've looked primarily at the integrase inhibitors alone.
Yeah, when we started these studies, I actually started with raltegravir. And it's at that time, and the field changes so rapidly, I feel, especially these days with the regimens that patients are taking, that it was important to us when we began to look at the combinations, because this is what the patients are taking.
Right.
It's and I do agree it's important to look at the individual effects, and I think that that is a timely comment and suggestion, because now, such a short time later, we're moving towards two drug regimen, moving back to two drug regimens, even giving single drug PrEP. I think lenacapavir's in trial for that right now, right? Yeah. So you're really underscoring Scott's point here about the importance of the individual drugs. The flux assays that we do, the turnover assays that we do, are challenging. That would make it, much, much bigger of an experiment to do the individual drugs. But we have a reporter for autophagy that we could use to probably more adeptly address the individual drug question.
Yeah, no, thank you. Like this really joins nicely our work, and I'll be in touch with you. I mean, I'll send you our latest publication, and the other one on autophagy is a very short report that is presently under review. So some of you may see it. I don't know. Thank you. Thank you
Thank
so
you.
much. Thank you, Laura.
Shilpa? Hi.
Great. Yeah. Hi, Laura and
Hi.
Joan. Terrific. Kudos to you guys. Great work.
Thank you.
We are also interested in autophagy, as you all know, and mitophagy. Just wondering, what do you think is happening at the end point with the cells? Are you thinking there's more activation, there's less of neurotoxic factors released, more EVs released, because that's linked to autophagy? I'd be curious to know, what do you think is
Yeah, fantastic question. I will be frank and say I don't think about EVs all that much. So I'm not sure I could intelligently or say anything about that. What we've thought about more in the past is the inflammation that might be happening. Astrocytes, as you know, are very important immune regulators, and autophagy is important for dampening down an immune response. And so
Yeah.
I think that would be a rich arena to look
I think
at.
that's a low-hanging fruit, and
Yeah.
worth looking
Yeah.
at. And great job.
Thank you very much.
Thank you.
Thank you so much. I apologize if I'm missing hands. There were 3 screens here, so if anyone else has a question, just
I think Dr. Perry Yasami, not to call him out, I don't know him, but he had raised his hands a couple of times, so I don't know if he wanted.
I saw his hand up too, yeah.
Hi,
Yeah,
Laura.
what's up?
This
Yeah.
is
Popped off my screen.
Swami.
Oh, there. Hi.
Excellent talk. Yeah, as Dr. Bush said, we are interested in autophagy, mitophagy. This is a very excellent talk.
Thank you.
I have a question. Did you check the others astrocytes like mouse astrocytes or rat primary astrocytes or any other cell lines in the similar phenomena?
No, we've not looked at rat or mouse, and these cells are not a cell line. These are primary human astrocytes.
It's a fetal or adult primary astrocytes?
The former.
Fetal.
Yeah.
Okay. Great. Excellent work.
Thank you. Thank you very much.
Thomas, question?
Yeah, hi. Caroline called me out this morning for being a grant recipient, so I figured I should say something. Yeah, really beautiful talk and really great work.
Thank you.
And I had to take a call while you were giving your kind of future directions conclusions page, but have you thought about adding rapamycin or an mTOR inhibitor to potentially try to decouple the effects of ART from this autophagy inhibition?
If I understand your question correctly, you're asking me about using rapamycin or other mTOR inhibitors or maybe even activators to understand better the effects on autophagy? Like how the inhibition is happening?
Correct. Yeah, I guess the question is kind of twofold. What is the mechanism of autophagy inhibition, and can you abrogate that effect by using like an mTOR inhibitor or some other
Okay,
agent?
okay, it's clear now. Thank you. So I don't know what the mechanism is. That is absolutely something I've been working on. I didn't show any data related to. I think there's two defects here, one in biogenesis and one in degradation. And to be honest, I haven't thought about how degradation itself might be inhibited yet. I've been focusing on the biogenesis. And I've done Western blots for mTOR phosphorylation, which was negative data, but I received some input about a year ago about looking at downstream molecules from mTOR to see if there's changes in phosphorylation there that might be contributing to the biogenesis. I've also looked at ETG5, and that was negative data. And I didn't go over this data here in this presentation, but LC32 accumulates after 24 hours of treatment with the ART, which is expected if you're inhibiting degradation. But that accumulation is not happening at the 7 days. And so I've wondered, is there an issue with LC3 transcription itself? I have yet to complete the QRT PCRs for that. We've not thought about rescuing to get to the other question. I think that's a great point, and certainly very important to sort of demonstrate perhaps even more clinical relevance about targeting autophagy and the impact that these drugs might have on the disease, as well as the implications it could have for improving the NCI outcomes. Did that answer your question?
Yeah, no, thanks for explanation.
Okay, thanks.
Shilpa, did you have another question, or you just didn't take down your hand?
Sorry, I need to
Did I
take down
get
my hand.
Any other questions?
Okay, Lara, maybe I have missed, or maybe you have done. Did you perform any LC3 accumulation assay or P62 degradation assay?
I'm sorry, say that again?
LC3 accumulation assay or P62 degradation assay, so that you can confirm whether the autophagic flux is due to the LC3 accumulation or defect in P62 degradation.
Okay, so I did both of those. I did turnover assays for both the LC32 and P62. I didn't show here that I did do P62 QRT PCR to make sure that changes in the P62 protein were related to autophagy changes and not transcription changes. There was no difference in P62 message at 8, 24 hours or daily for 7 days, confirming that P62 turnover is inhibited.
Okay.
Yeah.
Okay. Thank you.
Anybody else? Okay, well, thank you again to everybody for coming, and thanks, Laura, for such a great talk in science. And
Thanks, everyone.
next week.
Yeah, thank you so much. And thank you, Joan, for moderating as well. Thank you, Laura. Just so everyone knows, next week, Kristen McLaurin from University of Kentucky is presenting Development Gone Awry, Long-Term Neural Consequences of Chronic Adolescent Oxycodone Self-Administration. And then I also wanted to let everybody know, just remind people that you can register for the 2025 Symposium, JHCon. It's taking place April 8th from 9 to 3, and you can join virtually, or we're holding it in person too at the Owens Auditorium. So I've put the link to register in the chat, and I'll also follow up with another email. Okay, thanks everybody.
Thank you, everyone.
Thanks. Nice job, Laura.
Thank you.